officially recording here. So we're going to cover the integumentary system here. And this includes skin, hair, fingernails, toenails, and the mammary glands. Okay, so this is pretty much the largest single organ in the human body. And I just realized something with my video here. There we go. Got to get little old me here. The other cam, I have a USB cam hooked up to so we can look at a plastic model of this stuff in real time here, but not right away. Anyway, so essentially, it's going to weigh in at a little bit more than 2 kilograms, 4.5 pounds in the imperial or legacy measurement system. The liver is the biggest internal organ. The integumentary skin is not an internal organ. It's kind of the outside of us. You know, it's something that you drape over all the muscles and bones and other soft tissue stuff to keep the outsides outside and our insides inside. Okay, so if we consider this to be a single large organ, it's going to consist of obviously the skin and the layers, also hair follicles, fingernails, toenails, the breasts are associated mammary glands, cutaneous, and anytime you hear the word cutaneous, you think, surface of the skin, sensory receptors, and there's various types of glands like sweat glands, oil glands, things of that nature. So the main job of the skin or the integument is basically to keep water loss at bay, to prevent us from drying up and shriveling up outside on a hot, dry, sunny day. It would be like a grape turning into a raisin if we did not have our integument, our skin on us. So preventing water loss, keeping water inside the human body, we're going to lose some just through respiration, breathing, and stuff of that nature. But we can reduce evaporative loss with a protein called keratin. So keratin, as you see on this photomicrograph here at very low power, scanning power magnification, is going to be deposited in the outermost layers, as shown on this arrow here. This right here actually shows 1, 2, 3 layers of the skin and a lot of stuff in there. Again, this is one of our slides. This is one of my images. We have some very, very good microscope slides in our library, in our collection. So other functions of the skin or integumentary system, besides the most important one, which is water loss. Without the skin, we'd be dead in a matter, we'd die of dehydration in a matter of hours, minutes. Especially depending on the heat and the relative humidity of our surroundings. You know, you'd last longer in Houston or Atlanta or Miami than you would in Vegas or Salt Lake City or Victorville, because, you know, the water gradient is less steep in those places. However, in all those places I just mentioned right now, it's kind of hot this time of year. So the other function of the integument is evaporative cooling, which is a type of thermoregulation where we regulate body temperature. You release water, you sweat, perspire, and then that water evaporates off the surface, taking heat energy with it, leaving our bodies cooler. Chemical communication through what we would consider things like sexual attractant pheromones, also the emotional responses of blushing or blanching. But we'll cover that in a second. Protection of underlying tissues from excessive heat. You know, you're outside in the sun, soaking up the rays, or maybe you're just walking from the parking lot to the classroom or the store in a place like Riverside, San Bernardino this time of year, and it's like 102 degrees in the shade. You know, that sun will cook everything it touches. However, we have a layer of skin and hair to sort of protect from that. And we have fingers with a lot of sensory receptors in them, so we can obtain tactile information about an object. Is it soft and smooth and fuzzy like a kitten or a puppy? Is it coarse and harder like an older dog? Is it rough like sandpaper? Is it smooth like glass? Is it hot? Is it cold? Things like that. Involuntary nonverbal communication by blushing or blanching. You know, so for example, you know, you could bring home a smoking hot date when you're early in college and in high school, and worst case scenario, your parents are there, they like your date, and then they start showing her your baby pictures and little kid pictures where you were wearing, like, oh my God, it's so embarrassing, you blush. Or you could be talking smack about your supervisor or boss in the break room, and unbeknownst to you, they walk up behind you and hear you for a few minutes talking smack about them. You turn around and you turn white as a ghost, the blanching reaction. So those are chemical communication things there. Also, a very important one to our overall quality of life, synthesis of vitamin D precursors upon exposure to sunlight. Vitamin D deficiency is a big thing in America. Why? Because we're all working inside buildings on computers, most of us. We do not spend enough time out in the sun. Okay, so those are the functions. A lot of the integumentary system does a lot. And let's see, you know what? I don't have production of breast milk for nourishing infants. That's what the mammary glands do. So this list here is incomplete. Got to add that at the bottom there. Okay, but not to this version of this file. Got to do that and then re-upload it. So the integument is, it's a layered organ. The skin is a layered organ, and there's three major layers, and they all are composed of different types of tissue. Outermost layer here, I grew up calling it the epidermis. A European term is epicutis. It's composed of quite a few layers, like 100 or 200, depending on if we're looking at the palm of the hands or soles of the feet, of what we call keratinized, or you can use the term cornified, stratified squamous epithelium. The middle layer is known as the dermis, or in continental Europe, I've seen it referred to as the cutis, and that consists of mostly dense, irregular connective tissue. That stuff is tough. And then the deepest layer is basically made of fatty tissue, adipose tissue, and that's known as the hypodermis. The term I learned is hypodermis. Over in continental Europe, they may use the term subcutis. You can also call it the subcutaneous fat, and in the professional workplace, everybody's just going to call it the sub-Q layer or the sub-Q. You administer this sub-Q, you give this sub-Q. That basically just means you're going to have a hypodermic needle goes underneath the skin, and you make your injection subcutaneously. Now, this hypodermis, it can actually be very, very, very thick relative to the other two layers. It may even exceed their thickness combined. It's going to go all the way down to connect with underlying muscle tissue and underlying bone. I need to pause this recording because that annoying 13-year-old dog, 13-and-a-half-year-old dog wants to go outside. So let me pause here. All right, you mangy varmint, you're distracting me from work. Okay, yeah, got that annoying dog taken care of. One of the headaches of working from home is that it's not professional. You have to have family members and everyone on board, understand that you have to have a good work ethic and can't have family drama and stuff going on in the background while you're trying to do a lecture recording. And it's like, ugh. Need to rent. I wish I hadn't sold my office buildings. I could just have office space in one of those and do all of this stuff from there. Okay, so this is a prepared slide, one of ours, of the plantar surface of the foot. And this is the entire unmagnified image. I put this like on a white piece of paper and took a photograph of it with like I think it was a cell phone pic or a DSLR pic. And this is unmagnified. This image here, from top to bottom, I would say it's about 1.6 centimeters. So it's less than 3/4 of an inch across and less than 2 centimeters wide. So it's a little bit under an inch wide. That's the actual real life image size of what you are looking at here. So the layers are just, they pop out insanely beautifully here. They're so prominent, you can hold it up to the light, hold it to a piece of paper and see everything. You don't get that with all microscope slides of in the integument. So from top to bottom, we got the epidermis, the dermis, the hypodermis, and then we have deep, deep, deep underlying muscles here. And then right here, we have something extraordinarily interesting. We have what's called a Pacinian corpuscle, a deep pressure receptor. We'll see this bad boy over and over again in the semester. So this is a different image. Again, one of our microscope slides. The magnification power is scanning power. So all of your student microscopes, most any college you go to these days, if they've been updated in the last 10 years, will have what's called a scanning power objective lens. You look at something at only 40 power magnification instead of 100 or 400 or 600 or 1000. And you might think, well, that's less magnification. That sounds like it's not as useful, but you'd be surprised. Sometimes it's easy to have too much magnification. With microscopes, constantly using high power all the time or oil immersion all the time when you don't need to is like taking a old school Lamborghini or early Dodge Viper to go get Starbucks or groceries in the Home Depot, Starbucks, grocery store parking lot. You're going to be feathering the clutch, wearing out your friction plate in first gear in a parking lot with a car like that. Ask me how I know. I used to do it. It's just too much power for the application at hand. And this is low power here. This is like taking ordinary 4-cylinder sedan here, where you can idle a thing through the parking lot and let people cross the road in front of you, and you don't have to worry about feathering a clutch on a powerful sports car. Anyway, so you look at this scanning power. We can also see the layers beautifully from top to bottom, 2 millimeters. So that's less than 1/12. That's about 1/12 of an inch. So we look at this epidermis. I'm highlighting it with the cursor. Dermis, I'm highlighting that with the cursor. And then down here is hypodermis, sub-Q area. Okay, so let's deal with the epidermis first. We have a layer of stratified squamous epithelium indicated on this slide. And like I said before, in the case of the soles of the feet, plantar surface, palms of the hand, palmar surface, especially if you work with your hands, you do masonry, stucco work, auto mechanic, construction, any of the trades, you're going to work with your hands and your skin's going to be really thick. You could be looking at 1 to 200 layers of mostly dead cells. So this epidermis, this is where the waterproofing occurs. This is where the keratin protein is laid down. As we already know from when we talked about stratified squamous epithelium, there's like 4 to 5 layers in stratified squamous, and that's what the epidermis is made of, stratified squamous. So you can totally see the multiple layers here, just beautiful. They show up spectacularly. Since I already covered these layers in histology, let's just review them. So follow the cursor here close to this yellow line. We have the stratum basal, and then this lighter purple area is the stratum spinosum, and this dark burgundy purple area here highlighted with the cursor is the stratum granulosum, and then this whitish gray layer here is the stratum lucidum. It's lucid. It lets light through. And then we have this pinkish layer up here on the outermost part. That's the stratum corneum, or totally quantified layer. So this particular slide shows everything just beautifully here. Now, next layer in the integument is the dermis. So this is primarily dense, irregular connective tissue. Within the dermis, there's 2 distinctive layers. There's a superficial papillary layer. That's also known as the Latinized term that I had to learn was stratum papillarosum. And it has this bumpy up and down appearance. So, you can totally see it here. You see these little bumps, these little, look like little cave or mountain formations out in the desert somewhere, like in Arizona or Utah. This is the papillary layer here. And then look down here, I'm circling it with the cursor, has a different texture to it. It has a different view. I don't want to say a different feel, but visually and structurally, it's different down here. That's the next layer, which is the stratum reticularosum or reticular layer. Anyway, this papillary layer, you can see these little bumps going up and down here. This basically forms the physical connective tissue structure for what we call dermatoglyphs. Dermatoglyphs, you look at your fingertip up close and you can see fingerprints, palm prints, toe prints, those lines crisscrossing on the back of your hand. So ridges and lines on the hands and feet, especially fingerprints and toe prints, they act as what we call friction ridges, which kind of helps us grip things. You ever had slippery, sweaty hands, and you're trying to hold on to something and it's slippery, you put on gloves, or, you can wrap sandpaper around stuff, or, have a roughened texture handle. That rough surface improves our grip, our ability to hold on to stuff. Now, so on this image, I kind of have the entire dermis highlighted in between these two yellow lines here. And again, it can be tricky to see, but you can see a difference in the density and thickness and weave of the collagen fibers forming these two layers. Make sure I spelled reticularosum. Yeah, that's Microsoft spell checker does not like that word there. It's the Latinized version of reticular. But this layer is the tough structural portion of the skin that gives skin its toughness. Makes it harder for your skin to get punctured by, say, cat scratches and parakeets, lovebirds and little parrots biting you, and wrestling with dogs and using tools on the construction site. For cows, if you eat the stratum reticularosum of a cow, that's what leather is made out of. You can make belts, handbags, holsters, luggage out of stuff like that, and upholstery for cars. So again, pretty much all dense, irregular connective tissue. There's actually a lot going on in a dermis. It's kind of one of the more complicated layers here. So, hey, what do you know? I just said that. So we have these plastic models, which are like 700 times life size, that show all the things going on in a dermis. These are the nice new 3D models we have in our in SOS 103 here. So there's a lot of stuff going on in here. Before we go on to the dermis on this particular model, let's just look at this part right here. So what I'm circling with the cursor is characteristic of what's called thick skin. And that's found on the palm of the hand, soles of the feet. And then over here on this side where I'm circling with the cursor, that's characteristic of what we call thin skin. On the scalp, inside of your arms, back, parts of the body that aren't subject to abrasion and harsh rubbing in a day of ordinary, in the course of day-to-day work and grabbing and holding stuff. So you notice that thin skin, the stratum corneum is much thinner. We have hair follicles, whereas everything else, the stratum basal, indicated by, there should be a number here, and that number is number 1. Let's see, this is a high-res image, so I am going to blow it up, magnify it to insane levels. Yeah, there is a number 1 right here. Someone put it on by ink. Number 2 here is stratum spinosum. Number 3 is stratum granulosum. Number 4 is stratum lucidum. And then number 5 is stratum corneum here between thin and thick skin. Okay, and then we have these little bumps, which are called dermal papillae. Let me shrink this bad boy down again. And there we go. So let us move on to the next image. This is a different model. This is Somso brand model, shows all the same things and kind of the same depiction. It's just that it's much more expensive, much heavier to carry around, takes up more storage space. Okay, so a list of things found in the dermis include hair follicles, what are called erector pili muscles, sudoriferous glands, which include things like sweat glands, sebaceous glands, which are oil glands, blood vessels, and sensory receptors for touch and pressure. So they're all shown on this particular plastic model. We got hair follicles, different parts of hair follicles, a little sweat gland known as an eccrine gland, erector pili muscle, all that stuff. Okay, so here's the problem. You read the textbooks, you read the lab manuals, they say one thing. And then you look at microscope slides and you see things that are occurring in areas where technically they're not supposed to occur. I used to teach off of old textbooks that said, oh, hair follicles are contained, within the dermis. And then I look at actual microscope slides that show this stuff extending way into the sub-Q layer, the dermis. I'm sorry, the hypodermis. So that causes a lot of confusion. In my life, of course, I have a major background in real estate investment, property management, finance, commodity futures trading, forex trading, where mostly everything's governed by lies and half-truths and misdirection. And you learn to believe your own eyes first before you believe anything that's read to you in a textbook or shown on the TV by a very pretty talking head. So I'm going to have to say, sorry, folks, if I see stuff deep in a hypodermis, I'm going to say it kind of occurs in the hypodermis as well, even if the textbook says, oh, yeah, those are all up in the dermis. Maybe it's just a way of phrasing. A textbook says these things occur mostly in the dermis, and they omit the fact that they can occur way deeper than that as well. Okay, so anyway, we got the hypodermis indicated here with this little yellow line here. Okay, so eccrine glands. Let's deal with some of these items one at a time here. Eccrine glands are also known as sweat glands. And of course, they function in thermoregulation, evaporative cooling. They produce a thin, serous, salty fluid that spreads out. It goes through these pores here. So follow the cursor. I have a pore, a sweat gland duct. It exits the surface through a pore, a little opening. Spreads out on the surface of the skin. It evaporates and cools you off. So eccrine glands are a type of what are called sudoriferous glands. Sudoriferous glands secrete thin, watery, generally salty materials. Great way to lose water through dehydration, perspiration on a hot day. And again, they cool down the surface here. They have a shape which is described as coiled tubular. And I thought I had, good, I do have the diagram there. Okay, so this image here, I know there's a lot of textbooks out there that say, oh, sweat glands, they occur in the dermis. This is a very nice plantar palmar epithelium slide. And we have the epidermis up here, dermis right here, and then totally check it out, man, hypodermis down here. And now I'm gonna circle these big masses of cuboidal, simple cuboidal epithelium, one here, down here, over here, here, another one here, down there. Huge number of sweat glands or eccrine glands here. Palms of the hands and soles of the feet have a lot of eccrine glands in them for cooling. Okay, and they all look like they're surrounded by adipose tissue or hypodermis here, so what's up with that? It is what it is. Okay, so here's a textbook, something I just ripped off the internet, right-click and put it on here. The best way to describe the three-dimensional shape of an eccrine gland is like a coiled up garden hose, and then you got the end of the hose with your hose nozzle just hanging on a fence slather or something like that, or a gate. That's basically the best way to describe it in ordinary terms of day-to-day objects that we're all familiar with. The proper term is coiled tubular. It's a tube, like a hose, like a garden hose, and it's coiled up at the bottom. Okay, so you see a lot of these glands on the forehead, scalp, hands, feet, and places like that. Okay, so primary secretion down here with this filtrate from blood plasma. And then there's some absorption, recovery of sodium and chloride, salt basically. And what exits the pore is a less salty water solution for all intents and purposes. Now, we have a large version of this called an apocrine gland. And these are primarily found in the axillary, armpit, and pubic regions. And they're typically larger. This is the artist's depiction on the Somso brand model of an apocrine gland. Again, notice this is in the thin skin here with hair follicles and stuff. So the apocrine glands, some folks say they're actually kind of heavily associated with hair follicles, and wherever they have their ducts going up through the epidermis and then opening to the surface of the pore, usually it's right next to a hair follicle. These are way bigger than the ones we have on our microscope slides. Let's stay on this page here. So the apocrine glands, they secrete a different type of quote unquote sweat. It has chemical signals in it, pheromones, musky, funky odor, sexual attractions, things of that nature. Possibly fear pheromones and other things. There's always the old saying, I can smell this person's fear. And there is a heck of a lot to nonverbal communication that really isn't taught. I'm sure there's probably some courses you could take, I don't know, advanced psychology courses, stuff that is really good for you're playing a Texas Hold'em, and also if you're in sales and marketing, negotiating at the corporate or political level. It's always good to know about this stuff because, a good salesperson, a good marketing person, a good negotiator for a corporation, a good purchasing agent, they're going to want to have a lot of human psychology knowledge in their background and understand nonverbal communication. If you play poker at the Vegas casinos, it's kind of the same thing. You got to be able to pick up on all the cues that your adversary is giving you, unless they're really good and are not giving them to you. I tell you, I've made some of my biggest poker wins when I was like stressed out because I ate too much at the all-you-can-eat bar at the all-you-can-eat buffet at the Bellagio, and I was in pain because I had a tummy ache. So I'm just trying to be straight, poker faced at the table, and it's like, oh my god! And all the other players were thinking, oh, this guy has a crap hand. No, I didn't have a crap hand. I had a tummy ache because I ate too much at the all-you-can-eat bar, and I had the nuts and just probably got about $700 or $800 off in that one round. That's pretty cool. Okay, so nonverbal communication. It's a thing, and it's important in real life. Okay, so let's go to the hair follicles here. These are again on the Somso brand models because they are labeled number 13 here. We have this sebaceous gland or oil gland. These glands will make your skin oily, make your hair greasy. We have this reddish orange structure, which is called an erector pili muscle. So erector pili muscles, in the Eldar days of yore, when I was a little kid, some of the classrooms in the poorly equipped schools had chalkboards. And in the old days, it was we had fingernails, we'd scratch a chalkboard with our fingernail and make that most horrible noise that'd make the hairs on the back of your neck stand up. Well, when your hairs on the skin stand up, that is called piloerection. Pilo means hair. So an erector pili is basically a muscle that makes your hairs stand up. Where they occur, they're attached to hair follicles, and the other side attaches just to the skin, the dermis itself. So cold temperatures. You go outside and it's cold, or especially if you get in a swimming pool, even on a day like today where it's kind of warm, you get in a pool, you get out, there's a light breeze coming across, especially with little kids, they're going to feel cold and their skin on their arms and shoulders and stuff will get all goose pimply, or chicken skin kini as we might call it. Cold temperatures, your hairs are being stood up in an effort to cut down on heat loss. It creates this boundary layer. Strong emotions. You're walking to the parking lot at night, or you're watching a scary, creepy YouTube video, and something just gets you a little bit scared. Not like, oh my God, we're all gonna die, panic scared, but scared is like, I'm gonna spin around and double tap you on the face, boy, if he keeps walking behind me on my six. Type scared. There's a phrase, the hair on the back of a person's neck stands up as a response to the presence of this creepy person, or that prank phone call, or whatever it might be. Strong emotions. Okay, so here on a microscope slide, this is not mine. I wish it was. It is beautiful. This one's off the internet. Beautiful high resolution image. We can see the sebaceous glands here, and let's see. Let me open this up, zoom in a bit. So this is simple cuboidal epithelium. What you see here, take a long look at this stuff. I don't see a lumen. I don't see anything around here indicating this is really epithelial tissue. It could sort of be mistaken for adipose tissue, but after you look at adipose tissue and see that you don't have these big round purple cell nuclei in there, you say, okay, that's not adipose tissue. This is what we call glandular epithelium, generally cuboidal, and this is big time sebaceous glands in here. So the three-dimensional shape of these things, it's like a bunch of figs or guavas on a fig tree or guava tree that bears multiple fruit. The fig tree I have in my yard just bears solitary fruit, but I had one, a smaller one, that died. And that one bore multiple little figs, and they're all clustered together like a little cluster of flasks. The term was compound acinar, that describes glands that have that shape. Okay, so acini is like Latin for flask. You buy that table wine at the Chianti, at a place, I don't know, Trader Joe's, Giuliano's sells it. It didn't come in your traditional wine bottle like you see it in most liquor stores in the booze section at Costco. It had this really elegant old world Italian flask shape, and that's sort of what the term acinus, acinar, acini describes. Okay, so what good is the material these things produce? I know it's oily, makes your hair greasy, makes your skin feel oily. If you don't clean it off, you're going to get zits, pimples, blackheads, whiteheads. They're annoying. However, sebum does protect our skin and our hair from damage of particulate, drying winds, ultraviolet radiation from the sun. So it has a protective aspect, a function to it, even though it's what we want to get out of our hair when we shampoo and clean up. Okay, now hair follicles, complicated they be. So first of all, they're layered structures, a lot of layers in there, and contemporary thought holds that they now go through 3 distinct stages of growth. And we're talking about scalp hair here. Anagen, which is formation and growth, 2 to 8 years. A single hair follicle on your head is going to be actively producing hair for about 2 to 8 years. And then it may stop growing for a couple of weeks, and that's a phase called catagen. Whoops, hold on a second. Let me make sure my video here is pinned there. There we go. And then telogen is a resting period where no hair is produced. That may last for 1 to 4 months. And apparently the follicle just sort of recharges, renews itself. So again, what I'm talking about here is scalp hair, but ordinary body hairs, pubic hairs, those can also do the same here. So these things, they have a definite life cycle to them. Okay, once you get old, like me, you start worrying about the ones that fall asleep and never wake up. It's like, you don't want to get bald. Guys generally get bald, but women can as well. As we age, these hair follicles just go into a permanent stage of catagen, and they don't wake up after that. So if you want to think of a hair follicle this way, you have a surface. You poke a hole into that surface with your finger, and you have this deep, skinny hole. And then you have the surface that follows the inside contours of this hole. That's essentially a hair follicle. It's essentially invagination, a hole of epidermis that goes way down into the sub-Q layer in many cases. So let's look at this. Let's follow in this image, which I just pulled off the internet because it was well done and easy to, for me to have to redo this myself. It would have taken a better part of a half a day, maybe a full day with no pay. So you follow this pink line, that's the stratum basal. We can follow it all the way down with the cursor, all the way down, then up, then down, and then back up on the other side. And then similarly, the next layer above that, the stratum spinosum, we can follow it a large direct part of the way down, so on and so forth. So the stratum basal, when it's in a hair follicle, we call it something else. We call it the stratum germinativum. Also at this point, I should say something. There are some authorities, some textbooks out there that say the word, let's highlight it on this screen here, stratum germinativum, the germinating layer, the germ layer that generates, can also be applied to the epidermis everywhere else on the body, where it includes the stratum basal and the stratum spinosum together, the two layers where the cells are actually alive and not dead yet. So there's that. Okay, now the cells of the stratum germinativum, as you see here, they undergo rapid cell division and then they transform themselves and essentially become hair. They get squished together, compressed into hair. Inner cells die and their entire formation, their cytoplasm, all the organelles, is replaced by that waterproofing protein called keratin. So hairs, nail, the epidermis, rhinoceros horns, deer antlers, stuff like that, goat horns, that's all keratin, protein. Okay, so this stuff gets pushed out of the hair follicle by continued growth and development down here where I'm circling it with a cursor. Okay, so the hair itself, we're going to have 3 layers surrounding it that we can see on your typical microscope slide. In a student collection at a college or university. You're going to have an outer an outer dead layer, non-living layer, and that's called the cuticle. So that's overlapping dead squamous epithelial cells that make the cuticle. And then you have the middle layer, which is the densest part, gives the hair its structural strength. That's called the hair cortex. Those cells are kind of on a dead side too. And then the innermost core is kind of pulpy and pithy. It's pretty weak. It's called the hair medulla. Okay, so ladies, you know, your hair gets long, you get those split ends, frayed ends. A split end is basically the cuticle just gets worn away from day-to-day existence, just living, and it exposes the middle layer, which then starts to fray and break, and then when the inner layer is exposed, the tip of that hair just falls apart from exposure to the environment and abuse and ultraviolet light damage, sun damage, if you want to call it that. Now, the hair follicle itself is also layered. To simplify things a bit, we have an inner epithelial sheath and an outer connective tissue sheath. So let's go back to this. So we have a nice 3D drawing of a hair follicle, transverse section here, longitudinal section here. So we have the hair itself is just right here in this darker circle on the inside, medulla on the inside, and then there's going to be a cortex surrounding that, and then a cuticle on the outside. And then we have this internal root sheath in orange, peach, apricot color. I'm highlighting it with the cursor. That's the internal root sheath, or I should say epithelial sheath, internal epithelial sheath. And then technically we have an outer epithelial sheath as well in purple. On the lecture notes on a previous page, I did not really differentiate that, but the stratum, the germinating cells, the actual live cells that are undergoing cell division are on the outside of this circle. Then we have this dark gray line here, which is kind of like basement membrane. And then we have our outer connective tissue sheath here in blue. So on my lecture, I just combine this apricot and purple layer here, orange and purple layer as the inner epithelial sheath, and then the outer connective tissue sheath is in blue. There's a lot of authorities, including the ones that made this diagram here, that will break down the structure of the hair in more detail. we can drown in details in this class. Same with physiology and micro. So I figured there's only so much we need to cover here. This I pulled off the internet. I wish it was my own image, but it's not. This is absolutely exquisite. First of all, adipose tissue. Look at these big white circles here. Classic fat tissue cells. And then we have blue staining connective tissue collagen fibers here. And then we have the purple and red staining epithelium. So we find this stuff in the hypodermis. You'll have a hair follicle originating way down in here in the hypodermis. Even if the textbooks say it originates in the dermis, well, this looks like hypodermis here to me, so I'm going to go with what my lying eyes tell me before I take the word of any book or any television show or any YouTube video or anything posted on the internet. Okay, so there is actually a lot here. So labeling that with a little bit of Microsoft Paintbrush, let's just start with this area down here labeled by D as in Delta. This here is called a hair papilla, like a little flickering natural gas blue flame-shaped structure. There's really microscopic blood vessels here called capillaries. They bring oxygen and nutrients to the developing epithelial cells down here at the base that are creating this hair. Now we got a problem. There are some authorities out there that call this a dermal papilla, and that I like to be a fairly tolerant, easygoing, inclusive dude, but at some point when you see absolute falsehoods being propagated that mislead students and make students' lives more confusing, you got to put your foot down and start opening up a can of whoop ass. This is called properly a hair papilla, not a dermal papilla. So let me go to my zoom controls, switch cameras, and deal with this. So I was able to score this little plastic epidermis model off the internet for like, I don't know, it was like $34, all things included, whereas the schools are paying like, I don't know, $1,600 plus for the SOMSO ones and several hundred for the 3B models. And plus they're small, lightweight, and cheap, and they store easily. You can't go wrong with that. So let's make sure I have the camera here in sharp focus. So dermal papilla, you know what? I'm going to dim the light. See, when I redid this particular house, I put recessed ceiling cans all over the kitchen, living room, dining room, family room area, and hallways and bedrooms even, and it's not the best lighting for videography here. So right here, hair papilla, bulb of a hair follicle, hair follicle. So this is the hair papilla. But you see these little bumps right here, and here we go. Let's zoom in, let's focus that. These little bumps are made by the stratum papillarosum of the dermis. And these bumps form the basis for fingerprints and toe prints. So these bumps here, these are the dermal papilla, and that little structure there is the hair papilla. Okay, so you can feel these. They're actually bumpy and rough textured. If we were meeting in class, you could just run your finger over them and you get two modes of sensory input on this, tactile and visual. So that's something to be worried, something to be cautious of when you look at images on the internet. If you use Professor Google to bring up and I get information on this stuff, there's some problems. Okay, so D is the hair papilla. Now, anything else on here? Let's talk about this other stuff here. So, A as in alpha is the cuticle of the hair. B as in Bravo is the hair cortex. C as in Charlie is the hair medulla. E as in Echo is the epithelial tissue sheath. And F as in Foxtrot is the outer connective tissue sheath. Probably it's best to just say epithelial sheath and connective tissue sheath rather than adding inner and outer, because technically there's an inner epithelial sheath and an outer epithelial sheath. The arrow is kind of towards the middle of the two. And there's also an inner and outer connective tissue sheath. And the tip of this arrow is again sort of at the boundary of those two there. So there's that. Okay, so this is our SOMSO brand model, the old one, and OMG, I'm sorry, ZOMG and Ermagerd, just to top it all off. We could so turn this into a lab practical quiz worksheet thingy. So we got a nice erector pili muscle, a sebaceous gland, an apocrine gland, eccrine gland. Oh, look at these two dealy boppers here. Up here is a light touch receptor for like when you're petting a kitten or a puppy with really soft fur. This is called a Meissner's corpuscle. And down here is a deep pressure receptor, like when you're pushing on something really hard, like a really sticky doorknob, or you're giving your significant other a deep tissue massage with your thumbs and stuff. This is a Pacinian corpuscle or deep pressure receptor, which I've not really talked about yet. Oh, speak of the devil. Cutaneous sensory receptors. What they do, they're tactile in nature. So tactile refers to touch, and they relay touch information to the brain. So we have what we call free sensory or dendritic or naked nerve endings, also called nociceptors. And on this image here, I hate to say this, I need to expand this, not necessarily for you guys, but because I really can't see very good here with my old eyes. This model here does not depict any free nerve endings. They detect things like pain and temperature. Then we have right here a Meissner's corpuscle, also known as a light touch receptor. And these are inside the dermal papillae. And it's just a nerve ending that's wrapped by a few layers of connective tissue cells to make this little capsule, an encapsulated nerve ending. And then we have Ruffinian corpuscles are not shown on this model, so I'll not talk about them just yet. We have these Pacinian corpuscles down here, deep pressure receptors, way the heck down in the hypodermis. They have a zigzaggy coiled up nerve ending, and there's a lot of layers of connective tissue and what's called myelinated cells that wrap around this thing. Okay, so looking at this as a worksheet here, we have a sensory nerve here. I guess this will end, well, no, this is a sensory nerve going to a Meissner's corpuscle. Ooh, what's this? What layer is that? Epidermis. What layer is this? Dermis. What layer is that? Hypodermis, subcutaneous layer, subcutaneous fat, or just subcu. And let's see, we even have a nice little eccrine gland here. Okay, so let's shrink this image down. Otherwise, if I don't do that, it'll totally make this PowerPoint presentation glitch. So I simplified the diagrams here because everything in the textbooks, you know, textbooks cost a lot of money just to make and then ship over here. So there's a lot of cost involved in making them. And then you get the retail markup to you guys, the student, and they're many hundreds of dollars. And they're trying to save money as it is. If they broke things down simply like I do in my PowerPoints here, with all these simple diagrams, you'd be looking at an $800 to $1,000 textbook that's a couple feet thick and weighs like 35 or 40 pounds just to carry it with a lot of paper, a lot of ink. So And I just made these simplified diagrams. We have in pink, epidermis, and then in purple here, I made the stratum basal. And then in this sky blue here, we have the papillary layer of the dermis, or stratum papillarosum. And in this lighter sea aquamarine color, the stratum reticulosum, or reticular layer. Now we have a nerve ending, dendritic nerve endings going to all of these. They wrap around and coil. So inside these dermal papillae, we have little Meissner's corpuscles, and they're wrapped in these layers of connective tissue or collagen. And deep down here in the hypodermis, maybe there's a little projection of dermal tissue surrounding this coming down. You have a nerve ending and lots and lots and lots of layers of what we call myelinated tissue surrounded by an outside connective tissue capsule. So a simpler diagrammatic of these cutaneous sensory receptors. This is somebody else's microscope's photomicrograph, not mine, but we have a nice Meissner's corpuscle sitting in this dermal papillae here. Epidermis and dermis are labeled here. Lots of Meissner's corpuscles showing up beautifully in this image. This section of skin tissue is probably from a fingertip. You get yourself a paper cut on your fingertip. This is what would be showing in the size of the cut where that paper made that gash in your skin there in the epidermis. A lot of things in here, a little blood vessel here, some little sweat gland ducts down here or there, a lot of dermis, dense irregular connective tissue, a lot of stuff on this image. It's a nice slide. Now, this image was on one of our slides in our room, and this is a Pacinian corpuscle. So in dark red here, surrounding it, we have that connective tissue sheath. And then all of these things are layers of myelin laid down by a special type of neuroglial cell. And then way at the very center is the actual nerve ending, the neuron, part of the neuron cell itself. So this Pacinian corpuscle or deep pressure receptor just showed up. This is a spectacular example of one. Here it is way up close and personal. So right in the center here, we have our dendrite of a sensory neuron. And then we have the nuclei of these Schwann cells that make this layer of myelin, concentric layers surrounding it. Okay, so that's pretty much it for that. This particular structure, let's go back way up where I saw it first. It's actually right here. This is it right here in this image. So it's a respectably sized structure when you get right down to it. I suppose just for kicks and giggles, I can blow that up, expand it a bit. There it is right there. Okay, let me shrink this down a bit. So this particular presentation, we kind of covered both lecture and lab components. Since I have this rather comprehensive yet very inexpensive, competitively priced model here, might as well go over those structures on this plastic 3D representation. So this is really good. It shows a hair follicle from the outside, wrapped in that outer connective tissue sheath, sebaceous glands hanging on it. Shows a sectioned hair follicle, sectioned longitudinally with the hair papilla, the bulb of the hair follicle, and then the layers, the connective tissue sheath, the epithelial tissue sheath, the medulla, the cortex, and the cuticle there. Let's see, I might have forgotten to mention that this expanded base of the hair follicle that surrounds the hair papilla here is called the bulb. Now, the part of a hair that projects above the skin, that is called the shaft. The part of the hair that's underneath the surface of the skin is called the root. We have a little bit of a root exposed here, showing the layers in like an expanded detail here. We have a very nice erector pili muscle. Erector pili muscles generally attach to the base of a hair follicle near the bulb, and they'll go up and insert on the dermis here, or I should say originate on the dermis and attach to the base of the hair follicle, and they'll pull on it, when you're really, really cold. For future reference on any and all plastic models we have, these red things that look like little pipes and vessels, those are generally going to be arteries carrying oxygenated blood, and their blue counterparts here are typically going to represent veins carrying deoxygenated blood or deoxygenated blood back to the heart. Let's see, nice little cross-sectional view of a Pacinian corpuscle here. I know it's that same light tan green as the hair follicle, but it's different in structure here. Let's see, rotate this to the side. Oh, this is nice. We have epidermis, dermis, hypodermis represented here. On this model, the yellowish stuff down here is hypodermis. Let's see, what else do we have here? We see cutaway view. So dermal papillae from the dermis. And then I got to hold this really still in the camera here, keep it centered. Stratum corneum on the outside, and stratum lucidum, stratum granulosum, stratum spinosum, and then stratum basal, this light turquoise colored layer in between the papillae of the dermis and this grayish white material here of the stratum of the spiny layer. Okay, this also shows these nice little Pacinian corpuscles attached to yellow nerve endings as well. FYI, if this was a real specimen of a human skin tissue, these nerve endings wrapping around this base of this hair follicle, this is what we call a root hair plexus. And that technically would not be attached to the same nerves going up here. These, I guess, are representative of a free nerve ending, naked nerve ending, dendritic nerve ending, or nociceptor. So pain and temperature, light touch, and ever wonder a good argument for why we have body hair on our arms and back of our hands? If a mosquito or some biting insect were to walk on there, we feel it because they brush against these hairs, and you brush against a hair and there's a root hair plexus here. Well, that mosquito, which could cause you grievous bodily harm if it's carrying parasites like malaria or something, alerts, basically these hairs alert your brain to the presence of that mosquito, and you can kill it on the spot. Okay, so that's it for that different view of Pacinian corpuscles here. And more free dendritic nerve endings. Notice that we have these little loops of blood vessels, tiny little capillaries in the dermis, providing nutrients, oxygen, sugar, amino acids to the energy demanding stratum basal and stratum spinosum of the epidermis here. Okay, so that's it for this lecture. A little bit of complexity to the integument. I will talk more about mammaries and breasts when we talk about female reproductive anatomy towards the end of the semester. There's a lot of complexity in those as well. Okay, so this is a cutoff of material for the first exam. Exam number 2 is going to be bones. And I will end this right now and get it downloaded, edited, and then uploaded in short order.